All right, so now we're going to finish off translation number 2. We're going to look at the 3 stages of translation, first in prokaryotes, and then we'll delve into eukaryotic translation, and then finish off with some drug targets for the ribosomes. So initiation in prokaryotic translation involves the use of initiation factors, and these initiation factors are typically called IFs, as you'll see in the figures. Initiation factors, and they are important because they aid in the assembly first of the 30S initiation complex and then of the final 70S initiation complex. Okay, and these initiation factors are part of the family of the P-loop NATPases, or quite similar to the G protein family. And again, they are able to undergo conformational, so substantial or significant conformational changes on nucleoside triphosphate binding and hydrolysis. Okay, and they're particularly important because these proteins serve as a molecular switch for the cycle between a GTP bound form and a GDP bound form. Okay, so this is basically a figure depicting the entire process for the assembly of the 70S complex. So here's the 70S over on this side. And first, as you see, we'll assemble the 30S initiation complex, followed by the addition of the 50S, and then we are ready to carry out protein translation. All right, so the key thing to remember that during protein translation, although our RNA is paramount in the process of translation, protein factors are also required for the efficient synthesis of a protein. Okay, so let's first assemble the 70S ribosomal initiation complex. For the first sequence of events in polypeptide chain initiation, what we need are 3 protein factors, the initiation factors, which are essential. And they include IF1, let me just write it here, IF1, IF2, and IF3. Okay, so the 30S subunit first forms a complex with IF3 and IF1. And binding of the IF3, which is over here, prevents the 30S from prematurely joining with the 50S to form a dead end 70S complex, which is actually devoid of mRNA and the formal met tRNA initiator. Okay, so you need to have the binding of the IF3 over here to prevent the premature joining of the 50S. IF1 then binds near the A site, so near A site, oh sorry, I have it depicted over here as well. Okay, and this helps direct the formal met initiator tRNA to the P site. Okay, so this will help direct this component to the P site. Okay, this is our special tRNA, so our tRNA formal met initiator, formal met. Okay, that's coupled to the formylated or charged to the formylated methionine. Okay, all right, so IF2, as you can see here, is also a member of the G protein family, binds GTP, and the concomitant conformational change enables IF2 to associate with formal met tRNA and that initiator factor. Okay, so we have these 3 that are critical that will assemble the 30S initiation complex. We have IF3 that binds to prevent the premature joining of the 50S. We have IF1, which binds near the A site, and this helps direct the initiator tRNA, which is depicted here, into the P site. And then we also have that initiator tRNA that's bound to the IF2. And then it's also bound to GTP at this stage, at that 30S initiation complex when that is formed. And remember, we also have the critical base pairing between the 3' end of the 16S rRNA and the Shine-Dalgarno sequence, which is depicted here in the mRNA transcript. So this is our mRNA transcript. And that helps establish the correct reading frame. So correct reading frame. All right. All right, so now we got our 30S initiation complex, and we're going to then assemble the 50S to it. Okay, so here is our 30S initiation complex from the previous slide. We see that the IF2 GTP-bound initiator tRNA complex binds the mRNA at that initiation codon. And this is, again, correctly positioned by the Shine-Dalgarno sequence interaction with the 3' end of the 16S rRNA. And we have the 30S subunit together to form the 30S initiation complex. Okay, so the next step in this process is that IF2 will stimulate the association of the 50S complex, 50S subunit to the complex. The GTP-bound nucleotide that is found in IF2 is then hydrolyzed. So you get GTP hydrolysis to form GTP plus PI. So that's released from IF2 upon hydrolysis. And it releases IF2 from the 30S initiation complex. And the result is the formation of the addition of the 50S, release of IF1 and IF3 to form the 70S initiation complex. Okay, so when these structural changes happen, it leads to the ejection of IF1 and IF3 as well when we get that GTP hydrolysis by IF2. So when the 70S initiation complex is formed, which we see here, the ribosome is ready for the elongation phase of protein synthesis. And we can see that the F-MET tRNA molecule occupies the P site on the ribosome, and the other two sites of the tRNA molecules in the A and the E site, which is over here, are empty. All right, so now we're going to focus on the second phase of protein synthesis, which is the elongation cycle. It is a cycle because we keep inserting amino acyl tRNAs to elongate the polypeptide chain before we stop protein translation. So it typically begins with the insertion of an amino acyl tRNA into the empty A site on the ribosome. So the amino acyl tRNA is depicted here. So it's inserted into the A site. Okay. The particular species inserted depends on the mRNA codon in the A site, and again, if that corresponds to the anticodon that I just highlighted here for that particular tRNA. The cognate amino acyl tRNA doesn't just simply leave the synthetase and diffuse into the A site. Rather, it's delivered to the A site in association with a 43 kilodalton protein called elongation factor 2, or EF2, and that's depicted right here. So EF stands for elongation factor, and then a 2, hyphenated 2. EF2 is another member of the G protein family, and it requires a GTP. So if you can envision that it's bound to GTP, this actually facilitates the binding to the amino acyl tRNA. All right, so the question is, why do we need an additional protein to actually bring in the amino acyl tRNAs? And why is it binding to EF2? And there's a couple of reasons for that, because it's known that EF2 actually protects the ester linkage in the amino acyl tRNA from hydrolysis. So it would be counterproductive to charge your tRNA and then have it to undergo immediate hydrolysis and removal of that specific amino acid. So it acts like a protector for that very sensitive ester linkage, which can undergo hydrolysis. And then GTP hydrolysis in EF2, remember this is bound to GTP, will only occur when the appropriate base pairing occurs with the codon and anticodon complex. So once we've established that we have the correct cognate tRNA and the amino acid that we need to incorporate into the corresponding polypeptide chain, then do we only get GTP hydrolysis. And again, there's a significant conformational change. So conformational change in the protein that allows to contribute to the fidelity of protein synthesis. So if you get the correct matching between the codon and anticodon complex, so there's a misincorporation, you don't get GTP hydrolysis and you don't get amino acid incorporation into the polypeptide chain. A key thing to remember is that EF2 will not interact with that initiator tRNA, so the one that carries the formulated MET. It will only interact with the internal methionine tRNAs, so tRNA MET, the ones that insert methionines internally, and all other tRNAs. And then conversely, IF2, which is bound to GDP, will only recognize that initiator tRNA carrying the formal MET. So you can write the I there to represent initiator. All right, so now we're going to cycle through the phase of elongation. And in the top figure, we actually see, again, our 7DS initiation complex with the initiator tRNA aligned with the initiation codon, all in the P site of the ribosome. Okay, so the second step in this process of protein synthesis includes bringing in the next amino acyl tRNA into that A site that corresponds to whatever codon is in the sequence of the mRNA. And again, it must correspond to the anticodon of the cognate amino acyl tRNA. We said that it is brought in with EF2 that's bound to GTP, so they associate, so you get that complex. Once the correct match is made between the codon and anticodon in the A site, as depicted here, we get GTP hydrolysis of EF2 such that the GTP is hydrolyzed to GDP. This leads the EF2 with a GTP bound. And again, the EF2 will then be regenerated by using EFTS, which again promotes dissociation of GDP from EF2 and allows for EF2 then to bind a new GTP so that it is ready to associate with another amino acyl tRNA. Okay, so basically the steps include the amino acyl tRNA being delivered to the ribosome and GTP hydrolysis causing the EF2 GDP complex to dissociate from that amino acyl tRNA. The inactive EF2 GDP complex is then recognized by the elongation factor TS, which is highlighted here. It's just a guanine exchange factor protein, and it promotes the dissociation of GDP. The EF2 EFTS complex will then bind a GTP, which causes EFT to dissociate, so come off, and we regenerate EF2 bound to GTP, which can then bind another amino acyl tRNA. So now we're set up with our initiator tRNA in the P site and the cognate amino acyl tRNA in the A site. So we can all predict what the next step in protein elongation is, which is the formation of a peptide bond. All right, so just coming back to the figure that we left off in the previous slide, we have our initiator tRNA with our amino acyl tRNA that's come into the A site. We then get peptide bond formation, so transfer of that formal met tRNA, onto nucleophilic attack onto the amino acid that is linked to the tRNA in the A site. And now we have an uncharged tRNA in the P site and our growing polypeptide chain. So growing polypeptide chain, which then starts to exit the tunnel or the channel, chain in the ribosome. Okay, so then we need to actually translocate the mRNA by one specific codon so that the next codon will become available in the A site for the next incoming cognate amino acyl tRNA. And this is actually accomplished by an elongation factor called G, or translocase. So the name is depicted here. Okay, and EFG is actually another GTP-bound protein. Okay, and what it does, it actually binds to the EF2 binding site on the 50S subunit. So it'll actually bind in here. Okay, and the stimulation of GTP hydrolysis by EFG causes a conformational change in this protein, and it forces the tRNA and the mRNA to move through the ribosome by a distance corresponding to one codon. So we need the EFG bound to GTP to bind the 50S subunit. We need GTP hydrolysis to cause that conformational change, which then actually translocates the tRNA and the mRNA to move through the ribosome that corresponds to one codon. So we go from this conformation down here, and then finally, when we get GTP hydrolysis, translocating the mRNA and the tRNAs by a distance corresponding to one codon. So we've actually moved everything this way in the direction, so it's gone this way. So again, remember the mRNA must move by a distance of 3 nucleotides, so the next codon is positioned in the A site to interact with the incoming amino acyl tRNA. And at the same time, the deacylated tRNA, so this one here, that's lost its amino acid, then exits or moves out of the P site into the E site on the 30S. And the peptidyl tRNA moves out of the A site into the P site. So then we'll have movement of this over here. So now our growing polypeptide chain is located in the P site. We've lost our deacylated tRNA. And when this occurs, the movement of the peptidyl tRNA into the P site shifts the mRNA by one codon, exposing the next codon that's to be translated in the A site. Okay, and then this EFG translocase will then bind another GTP and wait for the ribosome to be able to carry out peptidyl transferase activity and then translocate it again. And it's a cycle that keeps repeating, so that now you have your growing polypeptide chain that's attached to the tRNA in the P site, and then you can go on and bring in the next corresponding amino acyl tRNA that's brought in by AF2, bound to GTP, puts it into the A site. If the correct base pairing occurs between the codon and anticodon, you get AF2 GTP hydrolysis. Finally, peptidyl transferase activity, and then translocation of the mRNAs and tRNAs in the ribosome. All Alright, so the 3D structure of the ribosome undergoes significant conformational change during translocation, and evidence suggests that translocation may result from the properties of the ribosome itself. However, we did introduce these elongation factors, and these protein factors accelerate the process. And again, we know that EF2 is bound to GTP, and this helps deliver the amino acyl tRNA into the A site. EFTS will help exchange GDP for GTP on EF2, so it's a guanine nucleotide exchange factor. And then finally EFG, which is the translocase, helps promote the translocation of the mRNA. And the possible mechanism for this, again, it's hypothesized that the GTP-bound form binds to the ribosome near the A site in the 50S, interacting with the 23S rRNA in the 50S subunit. And the binding of the EFG to the ribosome will stimulate its GTPase activity of EFG. And so the properties and the sizes, they're not very large molecules, protein factors, but they're all depicted in this table, which of course by no means you have to memorize the numbers. You can always look up the sizes of these proteins in any biochemistry textbook. This slide, I just wanted to actually show you the actual structures of the EF2 complex with the amino acyl tRNA and that of EFG. And you can actually see that EFG actually kind of mimics the structure of the EF2 plus the amino acyl tRNA complex, in that it has that long region extending out that kind of corresponds to the L shape of the amino acyl tRNA, and then the component over here that kind of resembles EF2. And in that sense, it's able to fit very nicely near the A site in the 50S subunit. And upon GTP hydrolysis, this EFG protein undergoes a significant conformational change that displaces the peptidyl tRNA in the A site and moves it to the P site, carrying the mRNA and the deacylated tRNA with it. Okay, and then upon dissociation of the EF2, EFG, it leaves the ribosome and is ready to accept it leaves the ribosome, and then the ribosome is ready to accept the next amino acyl tRNA into the A site. Okay, so those are comparisons of the elongation factors in prokaryotic protein synthesis. Alright, so we've talked about protein initiation and assembly of the 70S initiation complex. We've been through the elongation phase and how it cycles using EF2, which brings in the amino acyl tRNAs, in EFG, or translocase, which helps translocate the mRNA and tRNA complex in the ribosome, a distance of 1 codon in length. So now we're going to finish off by looking at the final phase of translation, which is termination, much like how we saw with protein transcription. So amino acyl tRNAs don't normally bind to the A site of a ribosome if the codon is UAA, UAG, or UGA. And as you remembered when we talked about the genetic code, these all indicate a stop in the mRNA transcript. And this is because normal cells don't usually contain tRNAs with anticodons that are complementary to these stop signals. Instead, these stop codons are recognized by release factors, and are denoted using the acronym RFs, which are proteins that promote the release of the completed protein from the last tRNA. And in prokaryotic protein synthesis, we have 3 RFs, and they consist of RF1, RF2, and RF3. RF1 will recognize the stop codons UAA or UAG. RF2 will recognize the stop codons UAA or UGA. And while RF3 just actually mediates interactions between RF1 or RF2, depending on which one is bound, and the ribosome. Okay, so and the ribosome. All right, so again, protein synthesis termination, as we'll see, is just a simple hydrolytic reaction where we have water nucleophilically attacking the final carbonyl in the polypeptide chain. So this would be the C-terminal end of the protein and our N-terminal over here attached to that terminal adenylate in the tRNA, and release of the polypeptide chain. All right, so our stop codons are recognized by release factors. RF1 is specific for UAA or UAG, RF2 specific for UAA or UGA, and RF3 will just mediate interactions between either RF1 or RF2 and the ribosome. Okay, and RF3 is actually very analogous to EF2 in that it actually has GTP bound to it as well. This slide just actually depicts one of the release factors. So RF1 is depicted here, and again, it recognizes the stop codon in the A site and will stimulate the release of the completed protein from the tRNA in the P site. RF1 and RF2 are compact proteins, as you can see here, and they kind of resemble a tRNA molecule, but you can see in this region up here, they actually contain a water molecule, which is responsible for that hydrolytic reaction to cleave the finished polypeptide chain from that terminal adenylate on the corresponding tRNA. Okay, so in this figure, we can actually see that we finished translating our polypeptide. We have a full-length nascent polypeptide, and we've reached the stop codon in our A site, which is empty, so stop codon. We either have RF1 or RF2, depending on the stop codon sequence. That complexes with RF3, which is bound to the GTP in this figure. Okay, we'll then recognize that stop codon in the A site, so in the A site, and stimulate the release of the completed protein from the tRNA in the P site, so that we get the subsequent hydrolysis of the nascent polypeptide chain from that terminal adenylate in the tRNA. So although the precise mechanism of release is not known, the release factor may promote, assisted by the peptidyl transferase site, a water molecule attack on that ester linkage, freeing the polypeptide chain. This detached polypeptide chain then leaves the ribosome. Okay, so then we get dissociation, and it leaves the ribosome. Okay. Once we have release of that nascent polypeptide chain, the transfer RNA and the mRNA remain briefly attached to the 70S ribosome until the entire complex is dissociated in a GTP-dependent fashion. So we'll get GTP hydrolysis by RF3, and they leave. And then we also have a ribosomal release factor, RRF, so ribosomal release factor. And this is also bound to GTP that will cause dissociation of the entire complex, so that we get an inactive 70S ribosome with empty E, P, and A sites, and dissociation of the mRNA and that transfer RNA that was found in the P site. And again, this is carried out by hydrolysis of RF3 and also the ribosomal release factor. And this slide just basically summarizes the release factors, RF1, RF2, and RF3, found in prokaryotic cells to carry out protein synthesis termination. RF1, again, very small protein, recognizes UAA and UAG as the stop codons. RF2 will recognize UAA and UGA. And RF3 just bridges the binding of RF1 or RF2 with the ribosome. It will bind GTP and stimulate the RF1 or RF2 binding and recognition of the UAA, UAG, and UGA stop codons. Okay, so that basically summarizes the process of protein synthesis in prokaryotic cells. Key thing to take away is we have initiation factors that help in the assembly of the ribosome in bringing in the initiator tRNA. We have elongation factors that will help bring in the corresponding amino acyl tRNAs, and elongation factor G, which is the translocase, to help move everything within the ribosome, so carry out translocation. And then finally we have termination factors, RF1, RF2, and RF3, that help stop protein synthesis and release the nascent polypeptide chain from the ribosome to then go on and carry out its particular function within the cell. All right, so here we're going to pause and actually try to answer the question of how many ATPs and how many GTPs would you require to synthesize a protein in E. coli that is 9 amino acids in length. So hopefully you've been keeping track of all the proteins and that require GTPs during protein translation, and in particular how many ATPs we actually use in this process as well. So we're going to walk through this step by step. You might want to pause the video here and actually try this yourself and see if you come up with the same answer that we will go through. Otherwise, you can just follow along. So the first thing we need to do when we're going to synthesize a protein, of course, is going to be to charge our tRNAs. So charge our tRNAs, and so that we make them amino acyl tRNAs. Amino acyl tRNAs. Whoops, tRNAs. And if you remember back, we said we need at least 2 ATP equivalents to charge each tRNA. And why do we need 2? One is basically for the synthetase, and then the other is for taking the AMP, amino acyl adenylate that's formed and then released upon charging of the tRNA. Remember, we get AMP plus PPI. Take another ATP plus that AMP to make 2 ADP for the ATP synthase. Okay, so for each tRNA, we need 2 ATP equivalents. So if we're making an amino acid that's 9, a polypeptide that's 9 amino acids in length, we have 2 times 9, which gives us 18 ATPs to make 9 amino acid protein. Okay. Again, the 9 is coming from 9 amino acids. All right, so then in the process of translation, we're going to break it down so that we do initiation first. And remember, during initiation, the only initiation factor that requires GTP is IF2. Okay, and again, so that's bound to GTP, so we need one GTP. Whoops, we need 1 GTP there. For elongation and translocation, so elongation and translocation. We need EF2, which again is another protein molecule that's bound to GTP. And we also need EFG. That's the translocase, which is also bound to GTP. And remember, we've already brought in our first amino acid, the initiator, the informal initiator methionine. So now we only need 8 EF2s and EFGs. So we have an equivalent of 2 for each insertion of amino acyl tRNA and translocation. And since we have 8 amino acids left, 8 times 2 GTPs, so that gives us 16 GTPs to elongate and translocate so that we create our 9 amino acid polypeptide that will then undergo termination. So on termination, we need RF3, which is bound to GTP. So here we need GTP, 1 GTP. And then finally, we're going to disassemble the ribosome. Disassembly using RRF bound to GTP. And again, that's another one we need. So then we can add up how many ATPs we need and GTPs. So ATPs plus GTPs. So in charging all our tRNAs, we basically need 18 ATPs. So 18. And then if we add up our GTPs, so one for the first amino acid with IF2, 16 for the remaining 8 amino acids using EF2 and EFG. So that's 17. Okay, one for the termination factor, RF3, so that's 18, and then one for dissociation of the ribosome by RRF, so it's 19. So 18 ATPs and 19 GTPs are required to synthesize the protein in E. coli that is 9 amino acids in length. Okay, so try to work through that process. If you have any questions about it, go back and review how we charge the tRNAs, how many ATPs you need, and then all the protein factors that require GTP during protein synthesis, while breaking down the stages. And remember, because we've already brought in here our first amino acid, we only have 8 amino acids left over to carry out our creation of our polypeptide, and then finally termination and then disassembly of the ribosome. Okay, 18 ATPs plus 19 GTPs. All right, so we just finished talking about prokaryotic protein translation. So now we'll focus on eukaryotic protein synthesis, which primarily differs in the translation initiation process. Okay, and so protein synthesis in eukaryotes typically entails more protein components, and we saw this with transcription, than does with prokaryotic protein synthesis. And some steps are more intricate. Okay, so we'll just very briefly give an overview of what occurs in protein translation initiation in eukaryotic cells. The first step is that we have the assembly of a complex on the 5' cap, and this complex is called the E, which stands for eukaryotic, IF4F complex, and you can see that it's made of, it's a multi-subunit complex, and there's a key one here that I want you to focus on, the EIF4E, which makes interactions with that 7-methyl G cap, which is depicted here in red. Okay, And then we have assembly of the 43S ribosomal complex. And in this complex, we have our initiator tRNA with our MET and our EIF2, which is bound to GTP. So this is quite similar to what we saw with IF2 in the prokaryotic cell. Okay. A key difference here is that in eukaryotes, the initiating amino acid is MET rather than N-formal MET, so there's no formylation of the methionine in eukaryotes. However, as in prokaryotes, there is a special tRNA that participates in the initiation, and this amino acyl tRNA is typically called MET tRNA I, so initiator as well, okay, with our MET here. But again, major difference, there's no formylation on the methionine. And once we have assembly of this complex, so we can see assembly of those two different complexes in stage 1. Stage 2, we have the IF4F assembling at the 5' cap, and then addition of the 43S ribosomal complex, such that we have this this initiation complex, the 40S, that's been assembled, and this complex is basically driven by ATP hydrolysis. So ATP hydrolysis, which will scan the mRNA. So this is our mRNA until it actually reaches the first AUG that it encounters. Okay, once it does that, we will then have addition of the 60S subunit, so the large subunit, to form the 80S initiation complex. So again, key thing, the initiator tRNA is methionine, but it's not formal meth. And the initiating codon in eukaryotes is typically always AUG, but unlike prokaryotes, eukaryotes do not have a specific purine-rich sequence on the 5' side to distinguish that initiator AUG from internal ones. So instead, the AUG nearest the 5' end of the mRNA is typically what is selected. All right, so we have the 40S ribosome that is bound to the MET tRNA initiator. And again, this attaches to the cap at the 5' end, the eukaryotic mRNA, and will basically search the transcript. Remember, this is the 5' methyl G cap that's been added onto the transcript, and it will search for that first AUG codon by moving step by step in the 3' direction. So you can see the direction this way, looking for this initiation codon. The scanning process is basically catalyzed by helicases that move along the mRNA, and it's powered by ATP hydrolysis. And I think this represents one of the helicases, if I'm not mistaken. Okay, and once we get pairing of the anticodon of the MET tRNA initiator with the AUG codon of the mRNA, we get the signal that the target has been found. So here we have codon and anticodon complex pairing. And in most cases, in eukaryotic mRNA, there's only one start site, and hence it is the template for a single protein. So it gets back to the terminology of monocistronic. In contrast, when we think about the prokaryotic mRNA, you can have multiple Scheindahl-Garnell sequences, and thus start sites and multiple start sites. And it can serve as a template for the synthesis of several proteins. And in that sense, they're said to be polycistronic. Okay, and again, remember that one of the most important initiation factors is the EIF4E, E, which is known as the cap binding protein also. So the cap binding protein, the CBP, since it specifically binds to the 7-methyl G-cap at that 5' end of the eukaryotic mRNA transcript. All right, so once we've found once we've made the encounter with the initiation codon, the small ribosomal subunit then positions itself so that the MET tRNA initiator interacts with this initiation codon at the P site. So this is at the P site. Okay. And then the final step really in initiation in eukaryotes is the addition of the 60S, which is depicted up here, to complete the ADS ribosomal subunit, which is this structure down here. And when we do have addition of the 60S, all the initiation factors dissociate, and we also have dissociation of EIF2, which undergoes GTP hydrolysis. GTP hydrolysis. And remember, all of these changes or dissociation, disengagements are accompanied by significant conformational changes in the structure of these initiation factors. And we then have assembly of the final ADS ribosomal initiation complex so that we can begin protein elongation. And all of these initiation factors will then dissociate and then hydrolyze GTP. And then finally, we're left with the initiator, MET tRNA, and the P site, and assembly of the ADS ribosomal complex. And then we can begin protein elongation in eukaryotic cells. Okay, so this table just highlights the many more initiation factors that eukaryotic translation needs compared to what we saw with the prokaryotic process. And again, I want to highlight the importance of the interplay between them, which is much more intricate. And again, when we talk about these factors, don't forget the nomenclature. The E stands for eukaryotic, and then the IF for initiation factor. Okay, so eukaryotic initiation factor, that's where we get this acronym from. So EIF denotes eukaryotic initiation factor. The key ones I want to point out here is, we've talked about this one, EIF4E. That is essential for binding to that 7-methyl G-cap, whereas EIF2 will bring in the initiator tRNA into that 40S ribosomal complex. Remember, it's bound to GTP, similar to what we saw with prokaryotic initiation factors. And basically, it will help bring in that initiator tRNA. Again, we have EIF3 promotes the initiator tRNA and mRNA binding. And it also prevents us reassociation of the ribosomal subunits in the absence of an initiation complex. So, key thing about the takeaway message from eukaryotic translation is that typically the 5' end of mRNA is readily available to ribosomes immediately after transcription in prokaryotes. However, in eukaryotes, the pre-mRNA has to be processed and transported to the cytoplasm before translation is initiated. And that 5' cap, so that modification that occurs to the transcript in the nucleus, provides an easily recognizable starting point. In addition, the complexity of eukaryotic translation initiation just provides another mechanism for regulation of gene expression in eukaryotes. Another key thing that we don't actually see in prokaryotic mRNA transcripts is actually the circularization of eukaryotic mRNA. So basically, eukaryotic mRNA is circular, and this occurs because when we have that EIF4E binding that 7-methyl G-cap, the 7-methyl G-cap, it can actually interact with the poly A tail, which is depicted here, through two protein intermediates. And the first protein that binds, the protein actually first binds to EIF4G. So it's this one here that binds to or makes interactions with 4A, EIF4A. And then this is when we have the EIF4E bound to that 7-methyl G. And then the N-terminal of 4G actually binds another protein, the PABP, which interacts with the poly A tail. And the PABP is the poly A binding protein. So we have protein interactions with the N-terminal of 4G between 4G and PABP. And then this N-terminal will then interact with the 5' end via interactions, protein interactions with 4E. Okay, so the protein first binds to this 4G to EIF4G to EIF4E, which in turn binds to a protein that's associated with the poly A tail, the poly A binding protein, so the PABP. Thus, we have the cap, which is depicted here, and then the poly A tail that are brought close together to form circular mRNA. So if this is our transcript, we can see that it's just a circular structure now. The circular structure is hypothesized to facilitate the rebinding of the ribosomes following protein synthesis termination, and that can basically enhance the efficiency of ribosome recycling. Okay, so that's another key distinct difference between prokaryotic protein synthesis and eukaryotic protein synthesis. All right, so we talked about the differences between prokaryotic and eukaryotic initiation in protein synthesis. So that leaves translocation and termination in eukaryotic protein synthesis. And basically, the processes are quite similar to what we saw with prokaryotes. There are effectively counterparts that carry out the bringing in of the amino acyl tRNAs and translocation. For EF2 and EFTS, which is the guanosine exchange factor, we have corresponding EF1A, and that corresponds to EF2, whereas EF1 beta gamma corresponds to EFTS, which is the guanosine exchange cofactor. So they will bring EF1A will bring in the amino acyl tRNAs. For translocation in prokaryotes, we had EFG, the corresponding elongation factor. In eukaryotes is EF2, and that also is bound to GTP, which will drive translocation after we get peptidyl transferase activity. And then finally, a very distinct difference in eukaryotic protein synthesis termination compared to prokaryotic processes. There's only one single RF factor. It's called ERF1, and it's responsible for recognizing all 3 stop codons. So major difference, there's only one RF compared to the 3 that we actually saw in prokaryotic protein synthesis termination processes. All right, as we now end off the course, we'll finish off with looking at some antibiotics that actually interfere with protein translation. We did talk about antibiotics earlier on and how they can affect transcription processes, and we had mentioned rifampicin, which was very specific for bacterial RNA polymerases, so blocking the RNA-DNA hybrid channel where we have the transcript exiting. And then we had also talked about actinomycin, which basically intercalates with the DNA to prevent RNA polymerase binding. And at high concentrations can affect DNA replication, so shutting that down as well. So antibiotics are known to inhibit protein synthesis. This occurs at the ribosomal level. And this table highlights some of the commonly known antibiotics. Some are very specific for prokaryotes, as you can see. Streptomycin and aminoglycosides. Prokaryotes, again, tetracycline, chloramphenicol. For cyclohexamide, we have something that's very specific to eukaryotes. So this is actually very toxic to us. Okay. Erythromycin, again, is very specific to prokaryotes. And we're going to focus on this one. We don't have time to actually look at all of them, but pyrimycin actually can inhibit or cause premature chain termination by acting as an analog of the aminoacyl tRNA. And this can occur in both prokaryotes and eukaryotic cells. Okay, so again, this table is just for your reference, but again, we can see the different ways these antibiotics can affect ribosomal activity, and some of them being very specific for prokaryotes and eukaryotes. But we're going to focus in on the ability of pyrimycin to actually cause premature chain termination by acting as an analog of the aminoacyl tRNA. All right, so we have the structure of pyrimycin that's depicted over here on the right, and we're doing a side-by-side comparison with the structure of the aminoacyl tRNA. So if we think of this as the terminal A at the 3' end, so this is our tRNA. It's attached here. And remember our 3' hydroxyl that has our specific amino acid that's attached. We notice that there's similarities in that we have an amino group here, and also in pyrimycin. We have this terminal end region, kind of looks similar to what we see with that adenylate that's hanging off the tRNA, but then we also have an additional structure here. One thing I want to point out here, so again, it resembles the aminoacyl terminus of an aminoacyl tRNA. Its amino group actually joins the carbonyl group of the growing polypeptide chain, as we'll see in the upcoming slide, to form an adduct that will dissociate from the ribosome. And we can get peptidyl transferase activity because of this the ability of the peptidyl transferase activity to occur, but then because of this amide linkage that we see here, we actually cause cessation of protein translation in not only prokaryotic cells, but also eukaryotic cells. Okay, so let's see how pyrimycin can actually stop protein synthesis in cells, in both prokaryotic and eukaryotic cells. So if we have in the P site our growing polypeptide chain, pyrimycin can enter the A site. Okay, so here's the structure of pyrimycin. Here's that key amide linkage, okay, and then that extra component that's attached to pyrimycin. And you do get formation of a peptide bond using that amide group on the extra portion of pyrimycin. So you'll get nucleophilic attack on that carbonyl, okay, and transfer of the growing polypeptide chain onto pyrimycin. And again, we get the formation of a peptide bond, which we see here. Okay. And again, once this is in the A site, and you'll notice that because we have that amide linkage that I highlighted before, it's not conducive to nucleophilic attack if we were to actually translocate it and allow for another aminoacyl tRNA to come in, because we don't have that oxygen at that 3' hydroxyl. So no oxygen at that 3' carbon location on the ribose. Okay, so in essence, puromycin inhibits protein synthesis by causing this nascent growing polypeptide chain to be released before the synthesis is completed. And because the puromycin is an analog of the terminal aminoacyl adenylate, part of the tRNA, it actually binds very nicely into the A site on the ribosome. And not only does it inhibit the entry of other aminoacyl tRNAs, it contains that alpha amino group, like an aminoacyl tRNA, which you can use to form a peptide bond with the carbonyl of the growing polypeptide chain, which is depicted here. The product, which is the peptide, right? The growing peptide with that has a covalently attached puromycin moiety at the C-terminal end. So this is the C-terminal end of the growing polypeptide chain. And this actually then will dissociate from the A site because it's weakly bound. So it's weakly bound. Okay. And very soon dissociates from the ribosome, and hence you get termination of protein synthesis. So not only does it block protein synthesis in prokaryotes, it also blocks protein synthesis in eukaryotes. It's actually quite poisonous to humans. All right, so this is how puromycin can effectively interfere with protein translation in both prokaryote and eukaryotic cells. And just briefly, just wanted to show the structure of streptomycin. This is an antibiotic that's very specific for inhibiting protein synthesis in prokaryotes. You can see that it has a very highly basic structure. So you get lots of amino groups conferring that basic characteristic. And basically what it does, it will inhibit protein synthesis initiation. Okay, so initiation and cause misreading of the mRNA. And it just prevents the binding of that initiator tRNA carrying the formulated meth, and will effectively stop protein synthesis in prokaryotes. So again, this is the structure, very complex structure of streptomycin, which by no means you have to remember. Okay, so before we end off, I'm actually going to leave you with two compounds that are pretty toxic to eukaryotic protein translation. One of them is diphtheria toxin, and the other is ricin, which we'll see in the upcoming slide. So the disease, or the toxin diphtheria, is a major cause of death in childhood before effective immunization became available. And the lethal effects are really due to the protein toxin produced by the diphtheria bacteria, and it typically grows in the upper respiratory tract of an infected person. A few micrograms of the toxin is usually lethal. Okay, so it's a very serious toxin if you get it into your system. The toxin typically is cleaved shortly after entering a target cell into two fragments, the A fragment, and the size is depicted here, along with the B fragment. The A fragment is what actually catalyzes the covalent modification of an important component of the protein synthesizing machinery, and whereas the B fragment enables the A fragment to enter into the cytoplasm of the target cell. And the target of the A fragment is EF2. And as we mentioned, EF2 is the elongation factor that's responsible for translocating during protein synthesis in eukaryotic cells. Okay, so how does the diphtheria toxin really block protein synthesis at the molecular level? And it's essentially just a very simple reaction that's depicted here on the slide. So here's our diphtheria toxin, particularly the A fragment. And what it does is actually take the ADP ribose moiety that's found in NAD+, so this portion here, so the ADP ribose moiety, ribose moiety that's found in NAD+, so it uses that as a cofactor, and transferring it on to a modified amino acid residue that's found in EF2. And this modified residue found in EF2 is a histidine that is post-translationally modified. So post-translationally modified to form diphthomide, and you can see the structure of that histidine residue down here. Okay, so it gets all this extra modification added to it, and this residue again is found in EF2, which is the elongation factor that will be responsible for translocation in eukaryotic protein synthesis. Okay, so this ADP ribosylation process, so just basically adding this onto that modified histidine residue, leads to the formation of this structure here, which is an ADP ribosylated diphthomide residue in EF2. So if this is the EF2 protein, okay, it's responsible for translocation during protein synthesis in eukaryotes. Essentially it blocks EF2's capacity to carry out the translocation process. So blocks translocation of the growing polypeptide chain, and then hence protein synthesis ceases. And this accounts for its extremely potent toxicity if it's found in the upper respiratory tract of an infected person. Usually, again, you only need a few micrograms of the toxin for it to be lethal. And it's just a simple post-translational modification that occurs on a diphthomide residue found in the translocation factor EF2. And will block translocation during protein synthesis and lead to the cessation of protein synthesis in eukaryotic cells. And finally, we'll end off by talking about ricin, which I have its 3D structure depicted here. It's a very small protein of about 65 kilodaltons in size. It's actually found in the seeds of the castor oil plant. It's a very deadly molecule, typically as little as 500 micrograms. So 500 micrograms is lethal to an adult human, and a single molecule can actually inhibit all protein synthesis in a cell. Its structure, as you can see, is heterodimeric in nature. There's a catalytic A chain, so this is the A chain depicted in here in blue, and joined together by a B chain by a single disulfide bond. So we got a single disulfide bond connecting these two subunits. Okay, the heterodimeric protein is composed of a catalytic A chain, which is responsible for N-glycosidic activity. So basically it will cleave the N-glycosidic bond. And once it enters the cell, and the beta chain actually allows for the toxin to bind to the cell, thus, and this binding leads to the endocytotic uptake of the dimer and eventual release of the A chain into the cytoplasm. Because it's an N-glycosidase, it actually uses the substrate, the highly conserved or universally conserved adenosine 4324. So it's very specific for this one here. That's found in the 28S rRNA, so in the large subunit. It causes cleavage of that glycosidic link, so you lose the base in that 28S rRNA. Once you remove this adenosine base, this 4324, found in position 4324, you completely inactivate the ribosome, and hence you prevent binding of the elongation factors, and this essentially will then shut down protein synthesis. So protein synthesis ceases. Because if you can't get your elongation factors to bind, you have essentially terminated protein synthesis in eukaryotic cells. I finally just want to end off by showing a similar cartoon representation of what we had shown in the first lecture of how we can interconnect all of the molecular biology machinery and tie it into the central dogma, where, again, if you recall, we had our DNA with our RNA polymerase that can synthesize a particular transcript. So this would be generated within the nucleus if it was a eukaryotic transcript, and then again transported out as depicted here, and then used in combination with tRNAs, which are also synthesized from the DNA and RNA to make up the ribosome. So they come together as these larger complexes. In the process of charging our tRNAs, use that mRNA with the ribosome to carry out the synthesis of a final product, which is a polypeptide chain. And again, these are essential critical proteins for cells to maintain cellular integrity. And hopefully you realize there are a whole slew of proteins that are required for these processes to work successfully. We talked about the synthetases that we actually need in the charging of the amino acids. We talked about the transcription factors that are essential to carry out protein transcription. We talked about all of the processing factors, so the guanylyl transferase, which will add on that 5-methyl-G cap, the polyadenylate polymerase, the spliceosome. So all of these factors, okay, that are essential. We have our rRNA, again, with lots of proteins to help in the formation of the ribosome. And again, assembly of the ribosome during protein synthesis requires initiation factors and elongation factors, and then RF factors for termination, such that they all facilitate and act in coordination to create nascent polypeptide chains. So I thank you very much for being in BCH 311 this year, and I wish you all continued success in your academic careers here at the University of Toronto.